Hi, we are going to review the neuromuscular junction model, and just a disclaimer, this one is my interpretation and your instructor may have a different one. What we're seeing here is a small part of a skeletal muscle cell, and the goal here of the neuromuscular junction is to get the muscle cell to contract, and by that we're talking about these structures here, which are called myofibrils. We see light and dark patterns here. Those are due to the contractile proteins that are found in a pattern in each of these. So we need to get these to contract. And to do so, we need to get a nervous impulse to this cell to get them to contract. And that's what we're seeing here. We're actually seeing the tail end, the axon of a motor neuron coming in here. This cell that we see wrapped around it 5 or 6 times is a Schwann cell, which helps to insulate the axon and speeds up the nervous impulse to get it to this muscle cell much more quickly than it would if it were not insulated. There's a material in here called myelin that acts as a great insulator, so the impulse moves very quickly. The impulse moves up the axon of the motor neuron and ends in these areas here called axon terminals. They're kind of bulged out. But if you look very closely, there's a slight gap between the cell membrane of the axon here in gray and the cell membrane of the muscle cell. The cell membrane of the muscle cell here is very convoluted. The structure is called a motor end plate, and it's specially designed for making contact with the axon of the motor neuron. But again, contact is kind of a misnomer here because there's a gap called the synaptic cleft. And so the electrical impulse coming in from the nervous cell needs to convert to a chemical signal, and then we'll convert it back into an electrical signal on the skeletal muscle cell. And so we have the synaptic cleft we have to jump. Turns out that all these little white dots here are called synaptic vesicles, and they contain a chemical called acetylcholine, which is a neurotransmitter, and its job is to jump that synaptic cleft. And so when the nerve impulse hits, bang, all of these synaptic vesicles will move toward the cell membrane of the nerve cell in gray and be released out into the synaptic cleft area. As the acetylcholine diffuses across, it will bind to receptors on this motor end plate. And again, this motor end plate is actually a piece of the cell membrane of this muscle cell, which is called the sarcolemma. As this motor end plate's a specialized part of the sarcolemma, and as more acetylcholine binds to it, chemical impulses will occur and we get charges moving across, and once enough acetylcholine is bound to the motor end plate, it gets electrically excited. When the motor end plate gets electrically excited, it will excite the rest of the sarcolemma going all the way up and down the cell. And this is your sarcolemma. Here it's more smooth because it's not the motor end plate, so we see it here. As that all gets electrically excited, that electrical excitement will pass into these blue colored tubes called transverse tubules. We see them here and see them across here. The transverse tubules are able to get electrically excited just as the sarcolemma is. You'll notice now also that these transverse tubules are next to this tan colored net-like stuff, which is called sarcoplasmic reticulum. It is a specialized version of endoplasmic reticulum, and its primary job is to house calcium ion. And you'll notice here on either side of the transverse tubule, you have a wide portion of that SR called a cisterna. And so we get what are called triads, which will be a transverse tubule and then 2 cisterna on either side of it. When the transverse tubule gets excited, it will trigger the release of calcium ion from the SR. Calcium ion will then go out into the myofibrils, causing them to contract. Just a few other notes here. All these little red guys are mitochondria, which will fuel both the motor neuron here and also the skeletal muscle cell. We also see cells out here. There's a satellite cell, which helps to act as basically a glial cell for the motor neuron, helping it out. This is also a nucleus. This is a nucleus of the actual skeletal muscle cell. And another nucleus here, we see the Schwann cell has its own nucleus. We can also make note of the sarcoplasm here. Usually if we're going to tag it during a test, we'll tag it in this area here, even though it includes all the organelles that are present. And I'm going to turn this around. just for my folks to note here that this structure here is called endomysium. It's actually a thin connective tissue covering, a areolar connective tissue that covers the entire outside of the skeletal muscle cell. And so that wraps things up so that we can show you how the impulse gets from a motor neuron to the axon terminals. We jump this synaptic cleft gap using acetylcholine, which is a neurotransmitter. It will excite the motor end plate, which then excites the entire sarcolemma. That in turn excites the transverse tubules coming in here. And when the transverse tubules get excited, we release calcium ion from the sarcoplasmic reticulum, and calcium ion causes the contraction of these myofibrils through procedures we will discuss in lecture.